Good afternoon, everyone, and thank you for joining us once again for this third webinar that we're running as part of the BNA's work to strengthen credibility in neuroscience, supported by the Gatsby Foundation. So I'm Madeline Lancaster. I'm a group leader at the MRC Laboratory of Molecular Biology, and I'm also a member of the BNA Credibility Advisory Board. And so we've decided to put these three webinars together as part of a focus on reproducibility and robustness on, in vitro models, and just kind of using this as an opportunity to discuss with leaders in the field best practices with these unique models. So this third webinar is featuring Clare Jones, who's CSO at Talisman Therapeutics, who will be telling us about human stem cell models of neurodegeneration. And she'll be giving us a bit more of the sort of industry perspective. So she'll be talking for about 15 minutes, followed by time for questions. So if you have a question, type it into the questions tab, and then I can ask them at the end. The chat is more for sort of general comments or issues that you might have. So try to keep your questions in the question tab, so I'll make sure I'll see them. And then I should also let everybody know that this webinar will be recorded. It'll be made available then afterwards on the BNA's YouTube channel, along with the other two talks that we've had last week and the week before, as part of this series, which I encourage you all to go and check out if you haven't seen them already. They're also excellent resources. So for those who didn't make it for the others, I recommend looking at that channel. And yeah, so I think with that, I've said everything that I need to say. So Claire, I think I'll turn it over to you.
Great, I will just share my screen.
Yeah. So I can see your screen, and now, yeah, I see it in presenter. Yeah, perfect.
Perfect. So I just want to thank Madeline and the organising committee for inviting me to take part in this webinar series. I'm really happy to be here to be able to tell you a bit about how we make our human stem cell models of neurodegeneration really robust, as well as being complex and relevant. Now, just to kick things off, data reproducibility is obviously fundamental to scientific progress. And when I did a bit of a search, I found this 2016 Nature survey where they questioned 1,500 scientists, not a huge number, but nonetheless found that in biology, more than 70% of researchers reported that they were unable to reproduce findings of other scientists. And about 60% of researchers couldn't even reproduce their own findings. Around 73% of respondents think that at least half the papers in their field can be trusted. Now, that's not a particularly high bar. And I think it's something that we all as researchers need to work on and try and improve. I think the results of data reproducibility problems are fairly obviously wasted time, wasted money. I mean, there's an estimate that up to $28 billion a year is spent on preclinical research that can't be reproduced. There's also the broader issues of loss of trust in scientific research and slower scientific progress. Working in the field of neurodegeneration, there are enough challenges without having to deal with data reproducibility issues. So this is something that I feel quite strongly about. So from a Talisman perspective, we use our human stem cell models of neurodegenerative disease as a platform to support drug discovery, all the way from target validation through pathway discovery, compound profiling, and all the way through to helping people make decisions about which molecules to take into the clinic. We use our disease-relevant model systems, which are a mixture of neurons, microglia, and astrocytes, all human iPSC-derived systems, and use these to answer a whole variety of questions, including mechanistic understanding of disease, compound profiling. We work with various different collaborators, small companies, big companies, but common to everybody is that they're looking for us to generate data that they can base decisions on. So it is absolutely fundamental to our work that our data is solid and reproducible, and that our collaborators can trust that they can take our data and use that to base decisions, which could then have significant financial implications for the company and certainly implications for the future of their projects. So in terms of how we develop robust experimental endpoints, this Nature survey had some suggestions on the key factors which contribute to the problems with reproducibility in research. From our perspective, what we do is spend time on the aspects that are not necessarily the most exciting, but really important. So we spend time on model development and validation, assay development and validation, and these are the two things that I'm going to talk about mainly today. We're also really careful on experimental design, being absolutely clear about what can and cannot be achieved within an experiment, very clear on data interpretation. We train all of our team members and we invest a lot of time in this. This will come back in almost every slide, but we spend probably about 6 months training all of our new recruits, and that is regardless of the level of background they have to start off with. We also standardize all of our protocols and continually monitor cell cultures and the data generated. And again, these are things which I'm going to come back to. So starting off on model development, I'm going to reiterate a number of things that were highlighted by Selena and Eva in the previous 2 webinars in this series. The first of all being the importance of quality control. So I think Selena said, quality control is boring but important. I mean, I put it's not exciting, but it's vital. It's pretty much the same message. We take a huge amount of care in QCing at every stage of our experiments. So we QC our IPS lines. We confirm that the mutations are present if we're expecting mutations to be present. We confirm the karyotype. We check that they're looking okay. We keep records at every single stage. Thinking of neurons specifically, we QC every single induction. So we have a set out process that includes using nanostring or qPCR for transcriptomic analysis. There's an example of this on the right-hand side. We basically have a panel of different genes, and what we're looking for is high expression of the cortical and excitatory neuronal genes, and low expression of, for example, interneuron genes. So in this example, the right-hand side block here would be inductions that we'd be reasonably happy with as cortical excitatory neurons. These are a bunch which I think we're aiming to be inhibitory interneurons, so it's not that we had so many that went wrong. We also would do that around about day 30. Then at a later stage, around day 60, we'll use immunofluorescence to look for key markers. So if we're aiming for excitatory neurons, we'd look at VGLUT1, VGAT, also potentially MAP2, and obviously DAPI. We also use bright field images really regularly to check how the neurons are looking. We make records at every single stage, so everything we do is tracked and monitored. And really importantly, we throw away the bad inductions. There's no point running QC if you're not actually going to act on it and ditch things which are not looking right. So secondly, on model development, we standardize all of our protocols. So we spend a lot of time developing robust protocols that work every time, and then we stick to them. So we standardize the protocols. We've got written standard operating procedures. These aim to ensure consistency in all aspects of cell generation and maintenance, and includes the need to keep comprehensive records of all the reagents. So maintaining supplier and product reference static as far as possible. The lot will obviously change, but that's noted. Also concentration. The established protocol is followed by everyone, and there are no exceptions. If any deviations are made because of things that are out of people's control, then a record is kept of that so it can be tracked. Changes are only incorporated when there's been a specific assay development effort aiming to do that. So obviously we do adapt our protocols if that's necessary, but only as part of a specific effort. I mentioned that we train all new team members. It's absolutely key that everybody in the team is working to exactly the same protocols and following exactly the same procedures. And this is continually monitored. So cells are monitored by the person who is culturing them, but also by the team members. and data is monitored continually to maintain standards, but also identify issues early, because there are things that are harder to control, and the earlier issues are identified, the sooner we can deal with them. So one thing that I think Eva highlighted last week was the need to regularly visually assess cultures. So here are some examples of images taken throughout neuronal induction from day 0 all the way through to day 25. The key point is mainly that things change. And I think as long as you know what you're looking for, you can spot to see whether or not you've got the cell morphology that you're expecting, that you're seeing these rosettes starting to appear, and that everything looks as you would expect. Some examples of where things didn't go so well. You know, I mentioned that if QC fails, you need to not use the cells. If you put rubbish into an experiment, you're likely to get rubbish out. So on the top of this slide, we've got some IPS that passed QC and were differentiated into nice looking neurons. On the bottom, some IPS that are clearly contaminated by some other cells. Now, in this instance, this induction was continued to see whether or not the contaminating cells could be removed. I mean, the answer at day 35 is not so much. And at this point, you've obviously got these big flat cells contaminating the neuronal culture, and these plates were discarded. Just a quick microglial example, very similar. We look morphologically at the cells. Here we've got nice morphology, good expression of IBA1 CD68, nice low level of TNF alpha release at baseline, good stimulation, past QC, these cells are good to go into an experiment. On the bottom, I struggled a bit to find examples because usually we just ditch cells that don't look good. So here's an image which is a little hard to see, but basically clearly doesn't look like these nice morphologies. There are a lot of flat cells. In this example, these cells were actually tested to see if there was an LPS response, and the answer was no. Just confirming really that if the cells don't look good to start off with, you're just wasting your time and money trying to get data out of them. Okay, so on to assay development. Now this is going to start getting repetitive because we standardize all of our protocols. We spend time developing robust protocols that work every time. On an assay front, we make sure that the assays are all validated, include positive and negative control treatments, and make sure that those controls are looking good with a good assay window. We standardize protocols and have written SOPs, or at least standard protocols, that ensure consistency in all aspects of the assay. So this includes cell preparation, so density, age, treatment, any other important factors. Timing, so timing of the experiment, timing of pre-incubations, timing of treatments, timing of the endpoints, because all of these things can have a huge impact on the data you generate. We also standardize media and reagents in the same way as we do for the model development. We keep records of all reagents and basically anything that isn't covered in the standard protocol. And these protocols are followed by everyone with no exception. So this is all kind of very reminiscent of the model development. Changes are only incorporated following specific assay development efforts. Again, we train all new team members and all team members who are new to a particular assay to ensure that they really understand exactly what needs to be done. And again, we continually monitor cells and data to make sure that standards are maintained and any issues are identified early. So to give you an example of why timing is so important, this is kind of quite striking, but really showing how for neuronal activity, it's so important to understand the time point that you want to look at, and also make sure that you're comparing like with like. So this is a graph which plots active object count, so neuronal activity effectively, using a calcium reporter in human neurons at different time points post-infection. So at 8 days post-infection, you got kind of low level activity, but nothing really synchronized. 17 days, you're starting to see some synchronization, and this really comes together at day 30. And actually starts to kind of reduce around by the time we get to day 58. So for example, if you were wanting to run an experiment and thought that the difference between day 30 and day 40 probably not make too much difference, actually there's quite a difference in the activity readout that you're seeing. And these are time-dependent effects. It's not anything to do with the treatment that you might be looking at. So it's absolutely key to make sure that you understand the activity you'd be expecting at a given time point and make sure you're comparing like with like. As an extra example, this graph shows the activity in two different media compositions, and there's clearly a huge difference. If I just showed you media 1, actually there is some activity, but it's very low level. You can see that in media 2, we've got really robust activity, and you can imagine that you would be able, in fact, we have shown that small molecule inhibitors can have an impact on this. So the spending time optimizing your conditions, including media conditions as well as time points, is really important. Finally, this is very brief and tries to cover a lot of information in a very small amount of time. We spend a lot of time on experimental planning and analysis. So looking at experimental design, we obviously include all the necessary controls, consider statistical powers and making sure there are enough replicates in each individual plate. We follow the established protocols that we've just talked about. We continue to maintain comprehensive records so that if there are any things that look a bit odd in data, you can go back and see whether there was anything strange that happened. We also make sure that key experiments are repeated to ensure data robustness. So going back to that Nature survey, we make sure that we can reproduce our own data. And as far as possible, analysis is standardized and automated, so particularly image analysis, to avoid bias. Unfortunately, I don't have time to tell you more about that, but that's quite an interesting topic in its own right. So to summarize, human iPSC-derived neuronal and glial models are complicated, but they can be robust and generate highly reproducible data. And we achieve that by spending time on assay and model development, quality control. We standardize protocols as far as possible. We plan everything thoroughly. We reproduce our own data. We make sure that we train all team members. And importantly, we make sure that if cells and cultures are failing the QC, we don't take them forward into experiments because you're just asking to generate rubbish data. The end result is that our data is trusted and our collaborators are able to use it to inform decisions that impact the future of drug discovery projects. So we feel that we're really making a difference to the projects that we work on. And that is the end. I should say that I haven't actually included an acknowledgement slide, but I've obviously taken data from various team members at Talisman. So my thanks go to them.
Great. Thank you so much, Claire. That was fantastic. Right, so let me just open up the Q&A tab. So if you do have a question, please feel free to type it into the Q&A tab. I have a few, but I want to give people a chance to ask some questions. There is one actually that just popped up. So this is from Mihai Lamora. Wonderful presentation. She or he is asking about sort of the media composition. Of course, I know you probably can't talk about what exactly is in there, but how sort of what you put on the cells really clearly from that slide showed a strong effect on neuronal activity. And so what's going on?
So there? I can say so. I'm not going to give the full composition, although we will hopefully be able to publish some of these data. Broadly, I forget which one worked. The one that looked good was that media 1 or 1, 2. From media, so media 1 is a kind of N2B27 based media, and media 2 is more of a brain fizz based media. That's in the literature already. Sure. So that's in line with what you would expect, but it was striking, and it did take a reasonable amount of optimization on various fronts. So media was part of it, but also looking at the lentiviral transfection of the neurons.
Right. Okay. So maybe I can just ask a more general kind of question about, you know, I mean, we've touched on this both, you know, in the previous talks from people in academia, but you've continued to touch on this, the importance of quality control. And I'm just sort of wondering what do you think is perhaps different about the industry setting when it comes to that? Sort of, are the standards different, do you think, than sort of what's going on in academia? Is that part of the reproducibility crisis as well?
Potentially. I mean, I think it's really widely understood in industry that it is absolutely fundamental that you're able to reproduce data. So you're never going to be able to get away with just running an experiment once and getting all excited about it if you can't reproduce it. We're working on systems that are aiming often to form the basis of assays that can then be used throughout a drug discovery program. So you have to show reproducibility, and I think that means that there's an acceptance that time and resource needs to go into those QC steps. That I can't really speak from an academic perspective, and Madeline, you'd be better than me, but my impression is that I think in industry there's more of an acceptance that time has to be spent there, and that's time well spent, whereas that's not necessarily the case in academia, where there's a much bigger push to get the shiny papers.
Yeah, so that's a perfect lead on to something else I wanted to ask. There are a couple of questions in the Q&A, and I will get to them. They're a little bit more specific. So I just wanted to ask you though, because it is clear, I think, from your talk that you spend a long time and, you know, setting up these standard protocols and making sure people are trained appropriately. So I'm just curious, kind of like, I mean, I know this is going to be different from each protocol to each protocol, but like, just to give us a sense of kind of how much time really does it take to standardize a protocol? Like, let's say you read something in the literature, somebody's just published something that looks like an exciting model that you might want to implement in the company. How much time does it take to get that going, let's say, if it does work at the end?
So I think if we're talking about, say, a neuronal system, because we've got the protocols for generating the neurons down, that means that we know our starting point is good. So then we're just we plug that protocol into the experimental endpoint. So if we're taking inspiration from the literature, then actually it's not necessarily too long, because at least we've got some good starting points. I mean, I guess a couple of months,
yeah, probably.
It is a bit variable. I think the slower bit can be really establishing exactly what media you're wanting to use. I mean, all of the activity work was run over probably about 4 or 5 months, working on many different things in parallel. But it's months rather than weeks, I would say.
Yeah, so exactly. So, I mean, because that does, illustrate also sort of that difference between, you know, industry and academia. I think a lot of academics sort of jump into these things and expect it to work right away, and I'm going to have data for my biological question, you know, once it reaches day 30, and that's it.
Yeah.
But, okay, so let me turn to the Q&A box. So there's an interesting one from Lindsay Marshall, who's asking, well, she's saying, so first of all, thank you for a really clear talk. And maybe these things are not exciting, but you explained why they are important beautifully, and I totally agree with you. So how much of the reproducibility issue does the standardization that you talked about actually address? Like, could we actually get to 100% reliability at some point? Or do you think we'll always have to kind of deal with this variability and do this QC and throw things out?
So I think you always have to QC, because I would never trust that just because you've standardized things, you don't have to QC. I mean, actually, that's the route to standards falling. I think if people get complacent that all you need to do is follow the protocols, we certainly find that once the protocols are established over time, actually the QC becomes much more routine. It's much simpler because everybody just gets better. And that's seen throughout industry with assays. It's a classic that your error bars get smaller and your Z primes look better as you've been running the assay for longer. So I don't think we'll ever get to the point where it's absolutely 100%. There's a question about, I think, reproducibility, whether it's desirable to get complete reproducibility. I think something along those lines was asked to Eva last week, actually, about whether some variability is desirable.
Right.
I think that's where it really depends on what the question is. So a lot of the work we do, there's a very specific question that actually it's not asking about human variability. It's asking about, you have a model system where you've got a phenotype that is disease relevant. And these are mixed cell populations. There's differences between inductions. It's never going to be 100% robust. We try and tighten it up as far as we can, but actually we're trying to make everything pretty reproducible so that we can say to somebody, yes, we think your compound maybe has an impact on the endolysosomal phenotype. I think if your question is how broadly applicable is this in the clinic, you probably need to go in the clinic. I think it depends on what your question is. And if you want to look at the variability across the human population, then I think, as Eva pointed out, that's really interesting. It's a huge study.
Yeah.
So those studies tend not to be the ones that we're looking at.
Yeah. So what about, there's a couple of questions here that kind of go in similar directions, sort of just how, you showed us that it's important to sort of throw away inductions that didn't work, and that those QC steps are important. Like, how much are we talking about throwing away? Are you talking about throwing away 50% of your experiments, or is it more modest than that? And also, like, do you see a lot of differences there when you start moving across different cell lines?
So I would say when I joined Talisman about 4.5 years ago, we would it depends a bit on the line, because some are easier. We would routinely throw away maybe 25% of the inductions. Now it's rare. I would love to say that's all down to me. Actually, I think it's more that as a group we have got much stronger on standardizing the protocols, and also most of the team have been with us for 3 or 4 years now, and they're just really good. So I think in the early days you throw away more, then you really get the protocols working to the point where it's not a brilliant standard protocol if you're having to throw away 50% of your inductions. I think that's where you need to spend more time on the development of the protocol. But when we're developing new systems, I think you do end up having to throw away stuff a little bit quicker, but we'd be aiming for ideally like 10% throw away.
So it's normal, I guess, when you're starting out, that things are not so efficient and they become more efficient.
Yeah, absolutely.
Okay, so maybe I'll ask one last question. There's a question sort of asking about when you're culturing these neuronal and glial cell types, and sort of how much they recapitulate the microenvironment of the living brain. And I also kind of wanted to ask, kind of related that, maybe more on the side of reproducibility. Are you sacrificing a little bit sort of the similarity to in vivo in order to get more reproducibility? Or do you think those things can go hand in hand?
I think it depends on the question. So we always try and be really clear on what question people want to ask. And I think if somebody wants to ask, so using the endolysosomal system again as an example, if somebody wants to know whether or not their compounds that have maybe looked interesting in an oncology model and are implicated in autophagy, if they want to know whether they have an impact on a phenotype that we see in neurons, then I would recommend they use a neuronal only system to test that, because we have the system set up and we can tell them whether it works. If they think that it's a neuronal mechanism of action, if they then want to kind of build on, well, what happens if you've got microglial, what happens if you've got inflammation, then that's brilliant. But I think if you don't need a complex system, I would go for the simplest. Obviously human, but I'd go for the simplest system to answer that question. If you want to model the living brain, then you probably want more cell types, and then its reproducibility gets tougher and variability increases. So I think it just depends so much on what question. Yeah, being really clear on what the question is. And, you know, we have lots of conversations about organoids. You know, obviously, Madeline, you're a better place than I am to talk about organoids. And for some questions, I think you need organoids, and for some you don't.
Yeah.
So it's picking the right model.
I think that is a perfect place to end. That is a really wonderful sort of final note. I just want to thank everybody who's joined us today, and I hope that you've gotten something out of this and the other 2 webinars. And like I said, these will all be available on the BNA's YouTube channel. And if you haven't seen the other 2, I highly recommend them. Many of the topics that Claire discussed today, we've also discussed previously in sort of slightly different perspectives. Yeah, thank you for attending. And thank you, Claire.
Thank you.
Okay, bye everybody.